And now page 2. Okay, so RNA is going to transfer the genetic code out of the genetic code is in the nucleus. So the genetic code is AKA the DNA. So it transfers it's not DNA anymore though, but it takes that code out of the nucleus and it takes it into the cytoplasm. So basically out into the world of the cell. And it's taking it out there to be translated by the ribosomes. So it'd be like, if you didn't know how to build Legos and you just had the instructions for the Legos, you would probably take that instruction manual to someone who was really, really good at building Legos. They'd look at those instructions and be like, oh yeah, I can build that. Here we go. It would then go out, grab their own Legos and start building your contraption. And that's essentially what ends up happening here. All right, so the ribosomes are one of the cell organelles, and here is its whole role. This is like its importance inside of the cell. The ribosomes use that messenger mRNA to create proteins. Okay, so how does that work? Well, a ribosome has two things we call two subunits. It kind of looks like hamburger buns, kind of. And so we have the top subunit here, and we have the bottom subunit over here. Now, we're going to have an mRNA molecule. Remember, it's a single strand. It's got the 5' cap. It's got the poly A tail. And then it's got the RNA sequence. It's going to enter the ribosome. And as it enters the ribosome, let's make a copy of this so that we can show what's going to happen here. So, this RNA is going to now enter inside of the ribosome. And as it goes through this passageway in the ribosome, there's the cap and the tail. As it reads each part of that DNA of the RNA sequence, this ribosome is going to read this part, and then it's going to go out and find this thing called an amino acid. It's going to read the next one, and it's going to say, oh, now I need this amino acid here. And then it's going to read this one. It's going to say, oh, I need this amino acid next. And it's going to read this one, and it's going to say, oh, now I know I need this amino acid as the last one. Okay, so as this mRNA then goes through, we are going to end up with, let me duplicate that. Okay, so once this RNA molecule is out of the ribosome then is going to release this thing up here. This now is our protein. Okay, so we have a ribosome. The mRNA goes through the ribosome interprets the RNA into a sequence of amino acids. Amino acids all together, amino acids are like little building blocks, like Legos. But if a castle is built out of Legos, you wouldn't call the castle Legos, you'd call it a castle. A protein is built out of amino acids. We don't call a protein amino acids because it's its own creation once it's been built. So now we call it a protein. Proteins. Proteins are the molecules that give organisms their traits. So if you have brown eyes, it's because you have a protein that gives you your brown eyes. If you have curly hair, you have a protein that produces your curly hair. If you have really long fingers, you have a protein that causes your fingers to be longer than average. If you have a disconnected earlobe, you have a protein that gives you that disconnected earlobe, and so on. So proteins are the things that give us, or any organism, their traits. So let's summarize. DNA. We start in the nucleus. In the nucleus. We have DNA. It lives inside the nucleus. It has to be protected. In order to get out of the nucleus, we have to transcribe that message into RNA. And remember, it's a messenger, so we call it mRNA. That mRNA then goes into a ribosome, And the ribosome is just that organelle that can transcribe, I'm sorry, not transcribe, but it can translate the message from an mRNA into a bunch of amino acids which are going to create proteins. So our mRNA is going to go into a ribosome. Ribosome adds a bunch of amino acids together to make a protein, and proteins equal, or they are the same thing as our traits. So the mRNA gets translated like we would translate a coded message into our language. Well, the mRNA is going to translate that coded message into a protein that creates traits. And so all of this happens in the cytoplasm, aka not in the nucleus. Yeah. All right, so if we have a sequence here, there's T's in it, so it must be DNA because RNA doesn't have thymine in it. So we know we have a sequence of DNA. Now what we need to do right now is we need to turn this DNA into RNA, and that process is called transcription, because we're going to transcribe the message, right? Like write it down into a new code. So we're going to create a sequence of RNA. Now, T on DNA means that our RNA sequence is going to have not an A. It would be an A if it was another strand of DNA, but it's not A because there is no, it would be A if it were RNA. T is going to build with A. Sorry about that. G is going to pair with C. C is going to produce a G on the RNA strand. T is going to produce an A. So again, another A. Oh, another A. C is going to create a G. T is going to create an A. And then the A, this is the one I was trying to get at earlier, is that there's no letter T here in RNA. That's not going to happen. So instead of a T, remember uracil is the new base pair in RNA. I almost got that mixed up there in the beginning. So now the RNA, we want to take our RNA strand and we want to create a string of amino acids, which is basically just a protein. So when you connect a bunch of amino acids together, we get a protein. This process is called translation, because remember we have a message, we need to translate the message. So the way that we do that is we look at the first letter, the second letter, and the third letter. So every 3 of these letters is going to create a new amino acid. This here is an amino acid table. You'll find this on the assignments, but you don't need to have this right now. For right now, let's just look at my amino acid table and figure out what our amino acid sequence is going to be. So ACG, A, C, G. That means that our amino acid is THR. Now I'm going to draw my amino acid here as a circle. THR is my first amino acid. THR stands for threonine, however you want to pronounce it. So our next one is AAA. So now we go A. So we find where those connect, and then AAA. Here we go. So now we have LYS. So this would all be done in the ribosome. The ribosome would connect THR to LYS because it's translating that RNA message. Okay, we have a string of amino acids building, so now we have starting to make a protein here. Let's do our last 3, GAU. GAU. Okay, GAU is going to code for ASP. And ASP stands for aspartate, or something like that. Now, this will continue on until actually we get a code on here that has a stop. So let's just imagine that these next 3 letters up here, if I were to change the color, let's just say that we had next a UAA. Those 3 would code for a stop because that is what the code is. It doesn't code for an amino acid, it just stops the process of the ribosome translating the message. So here we end up having our protein. This is going to create a protein. And remember, proteins are what give us our traits. So that's the process. We start with DNA. DNA goes into mRNA. mRNA goes through the ribosome. It's translated into a protein, and proteins are what give us our traits. This is the central dogma of cellular biology and genetics. That's all. Thanks for listening. There's your twofer.